Hi, Assalamualaikum everyone. This is CHM3202 Organic Chemistry. Today we are going to present about amino acid functions in plant development and their biosynthesis. First of all, my name is Muhammad Amir Azamuddin bin Abdul Ghaffar and my metric number is 200238.
Hello, my name is Nur Batrisha binti Hussein, metric number 204458.
Okay. So today we are going to talk about some subtopics, which are what is amino acid, types of amino acid function in plant, and amino acid and their biosynthesis.
So for the introduction, what is amino acids? Amino acids are actually molecules that combine to form proteins. Proteins will occur in every living organism and they have many different types and also have many different biological functions. For example, the keratin of the skin and our fingernails, the fibrin of silk and their spider webs, and the estimated 50,000 or so enzymes that catalyze the biological reaction in our bodies are all proteins. But regardless of their function, all proteins have a fundamentally similar structure and are made up of many amino acids linked together in a long chain, which we call peptides.
Okay, so what is amino acid? Amino acid are basic units for protein biosynthesis, other than being precursors of other molecules which are essential for plant metabolism, such as hormones, coenzymes, nucleotides, and polymer of the cell wall. These are organic molecules composed of carbon, hydrogen, oxygen, and nitrogen. Methionine and cysteine also contain sulfur. Amino acid is a difunctional molecule, which means that they contain both a basic amino group and an acidic carboxyl group attached to a carbon chain. Amino acids are building blocks for protein which play an essential role in the life of a plant. Proteins are responsible for the cell structure, but they also control all different chemical processes in the plant, from photosynthesis to transporting nutrients or regulating flowering. Under optimal growing conditions, plants synthesize their own amino acid through thousands of chemical reactions. The creation of proteins by linking specific amino acid initially into chains known as peptide also consumes a lot of energy. Reducing that energy cost allows it to be redirected to other aspects of plant growth and development. Under period of abiotic stress, plants decrease their production of amino acids and can even break down structural protein to recycle the required amino acid. A source of ready-to-use amino acid means the plant won't have to invest energy making them from scratch, and the need to break down proteins is reduced. So it will even have an additional supply of building blocks to help create the proteins required to counter the stress it's experiencing. This results in improvement of health, growth, and vigor. Okay, so what is the classification of amino acids? There are two classifications. First is hydrophobic, and the second one is hydrophilic. So hydrophobic is water-fearing, which is non-polar side chain. And one of example is alkyl side chain. So for hydrophilic, it is polar neutral side chain, and the example is anionic and cationic.
Now we continue with subtopic 3, which is the functions of amino acids in plants. So plants are a rich source of amino acids and their individual abundance in plants are of great significance, especially in terms of food. So the most abundant amino acid in plants are glutamic acid and aspartic acid. As you can see from the table shown, all three amino acids are used in protein synthesis, for example glycine. Glycine has a great chelating action and it improves the flowering and fertilization processes. Glutamic acid is used in chlorophyll synthesis and a promoter in the absorption of other amino acids. For tryptophan is a precursor of auxins and phytoalexin, as well as the activation of plant defense routes, for example, animal attack, mechanical damage, and drought. For methionine, used as a precursor to ethylene and polyamides, also used in the fruit ripening and color obtainment in plants. Plus, we have proline. Proline is used in the metabolism of stress. So now we continue with serine. Serine function as to regulate the amino acid resistant mechanism defense in the plant. So for example, it produces the defense for herbivore attack and mechanical damage. Phylanine is used as a precursor of certain antibiotics in some species, as well as an aid in the photosynthesis processes. Next, we have a set of amino acids which have the same function. For example, is leucine, lysine, tryptophan, histidine, phenylalanine, tyrosine, as well as glycine. So they are used in the alkaloid metabolism pathway, as well as the plant protection against pests and stress. Also for phenylalanine, used in the production of salicylic acid, as well as disease and stress prevention. And lastly, we have tyrosine, which is the precursor of plant defense mechanism. Or subtopic 4, which is the amino acid and their biosynthesis. So for this part, we have chosen lysine and glycine for the amino acid. So there are two pathways that have been identified in the nature for synthesis of lysine. So the first one is the diaminopymalate DAP pathway that belongs to the aspartate-derived biosynthetic family, which are also involved in the synthesis of threonine, methionine, and isoleucine. Whereas the second one is the A-alpha amino adipate, AAA pathway, is part of the glutamate biosynthetic family. So as we all know, our human body cannot synthesize lysine, but it's very essential in humans and can be obtained from the diet, for example, through our food. So for the organism that can synthesize lysine, which is plants, there are two main biosynthetic pathways, which, as I have said before, the diaminopymalate and the alpha amino adipate pathways. They both employ distinct enzyme and substrate and are found in very diverse organisms. So the lysine metabolism will occur through one or several pathways, and the common one, which is the saccharopic pathway. Next, for glycine, as you can see from the diagram, there are three very distinct pathways for the production of glycine in plants. For example, through the photorespiration processes, through the L-serine biosynthesis, as well as the L-threonine biosynthesis. So in plants, glycine can be generated from at least three routes. The first one involves the serine hydroxymethyltransferase. The serine hydroxymethyltransferase will catalyze a reversible reaction and the interconversion of the serine and glycine, which means it produces serine and glycine. So in plants, the reaction equilibrium will slightly favors glycine the reproduction. For example, in some parts of the plant, which is the leaves, during the photorespiration process, when high concentration of glycine is produced, the reaction is driven towards serine biosynthesis. And we have in roots and other non-respiratory photorespiratory tissues, the enzyme will more likely contribute to the production of glycine biosynthesis. Next is the second pathway, which is produced from the glyoxylate using glyoxylate aminotransferase, which is the middle pathway you can see from the diagram. So there are at least two distinct types of glyoxylate aminotransferase activities in plant. The first one has primarily serine, using serine glyoxylate aminotransferase activity, and the other will exhibit primarily glutamate glyoxylate aminotransferase activity. But overall, both activity will produce glycine. So for the last one is the glycine biosynthesis using the threonine aldolase, which will convert the threonine to glycine and acetaldehyde, which is the left part of the diagram. So now we can continue to the connections between the amino acid metabolism in plants. So you can we have three diagram, which is A, B, and C. For the A and B diagram, we'll conclude about the pathways, and the diagram C will inform us about the functions of the amino acid. So for the diagram A, usually starts from the aspartate amino acid, which will produce, for example, homoserine, lysine, glycine, proline, as well as leucine. So for the red arrow, it indicates the allosteric product, but via feedback inhibition for the production of the amino acid. And for the green, which is between the production of lysine to LKR, which produces saccharopin, it indicates the allosteric activities in the pathway. Next, we have the diagram B, which also start with aspartate pathway, but which produces different amino acid. For example, from 2,3-dihydrodipicolinate, it produces lysine, which has the function of starch regulation, unfolded protein responses, lipid metabolism, glycolysis metabolism, and nucleotide metabolism. So in diagram B as well has the set of amino acid which are used in the biotic and abiotic stress in plants, as well as producing the acetyl-CoA, which is important in the CA cycle or energy metabolism in plants, which means it produces energy for plants to function. So in diagram C, as I've said below, it explains about the functions of the amino acid that we discussed earlier. For example, proline metabolism in the biotic and abiotic stress, as well as for plant development, seed germination, root growth, flowering, pollen fertility, and embryo development, which is the plant seeds. And for the glycine, which is very important in plants for photorespiration in leaves, and the TCA cycle and energy metabolism, because chlorophyll produces energy for the plant's function. For threonine metabolism is for plant immunity against diseases, as well as stress and animal type. And then we have the alanine metabolism, also used in the TCA cycle, and for petatonate synthesis, lipid and carbohydrate metabolism, and polyamines metabolism. Arginine is also used in the biotic and abiotic, and also for the storage of nitrogen and mobilization. Next, we have the methionine, which is also consists of abiotic stress and mitochondrial energy metabolism. And that is all for our SCL presentation. Thank you.